So next we've got Professor Richard Grencis. So Richard is the lead for Pathogens, Parasites and Commensals. So Richard set up his own research group in Manchester in 1987 after a PhD in Glasgow and postdoc studies in Nottingham in immunoparasitology. And since moving to Manchester to set up his own group, Richard has actually been awarded the Chris Wright Medal by the British Society for Parasitology. So this is for outstanding contributions to parasitology. And this is actually the highest award given by the Society. So Richard had a special interest in parasites and specifically how they modulate host immunity. He's internationally recognised as one of the leaders in the field in immunoparasitology. So Richard's going to talk to us today about the research going on within the pathogens, parasites and also commensals branch.
Okay, thank you very much, Lizzie. So it's a pleasure, I think, to end up with the last talk this morning, so just keep with me for a few more minutes. But I want to talk to you today about some of the work that's going on in this particular branch of the Institute. And as a baseline, I suppose, I would argue that pathogens, parasites, and now commensals drive the evolution of the immune system. And therefore, understanding this complexity and the interactions between all these factors ultimately determine health in its broadest sense. Now, in Manchester, I feel we've got a real excellence in experimental immunology, in addition to the clinical expertise that we've heard about today, and particularly in this particular branch. And we're able to look in a mechanistic way to try and understand how the immune system is involved in these interactions. And through that, we extend knowledge, we learn a lot about the immune system and the pathogens and commensals. But ultimately, it will underpin translation. So Manchester actually has a particular focus on certain types of pathogens to define immunity, and now we want to look at it in the context of our commensals, the microbiota that we hear such a lot about. And we work with viruses. We have systems to work with viruses such as influenza, bacteria, for example. This is Citrobacter, a bacteria that infects the intestine. We work with fungi that we haven't mentioned very much today. So Aspergillus is a particular strength in Manchester and is a source of major allergens leading to lung disease. We also work with the so-called classical parasites, which are the eukaryotic organisms, single-celled organisms such as malaria, as shown here, an infected red cell. And on the right, we have a particular strength in helminths, which are the multicellular pathogens. And we're going to look at this in the context, ultimately, of the host intestinal microbiome, how these interplay. Now, we have a strength in Manchester in these large multicellular parasites. They've been regarded in terms of human infection as major neglected tropical diseases. And they're not neglected because they're not prevalent. They are extremely prevalent. Up to a third of the world's population currently will have, as Judy says, hookworms crawling throughout their lungs. And they've been with us throughout evolution. And of particular interest to us is the fact that they stimulate the immune system often in very different ways to the classical infectious agents such as viruses and bacteria. So we feel we can learn a lot about them. And in the context of our intestinal microbiota, we've been facilitated enormously, but as John mentioned, with the setting up of the Manchester Notobiotic facility, which allows us not only to look at responses in the absence of any microbiota, but to selectively put back certain good or bad bacteria, as we might think, and see what influence that has on immune responses. And it was very interesting to see that all the talks of this morning have also highlighted the importance of having good in vivo laboratory systems to work with, and that's Manchester Notobiotic facility is a part of our large and excellent animal facility in Manchester, and this allows us to examine right from mechanistic ways and translate them through to the clinic, and takes observations from the clinic back into a mechanistic status by looking at good animal models. And we couldn't do that without the facility, the animal facility, and all the other core facilities that the university has generously invested in. So now I want to go through a few examples to illustrate some of the work that we're doing and the complexity of it. And this is close to my heart, as Judy would say. And this, I'm going to start off talking about interactions between this particular parasite, which, as you can see here, is an adult parasite, is the whipworm. And this is a parasite that lives in the large intestine, and this is a section through a colon. And you can see, if you look carefully, the anterior end embedded in the epithelium and the fat back end out in the lumen. Of course, this parasite lives right in the site of our intestinal microbiota. And the interactions between the parasite and the host in terms of immunity and commensals is quite interesting. We've learnt a lot about how the immune system can control and get rid of these parasites, and also what goes wrong in terms of immunity that leads to chronic infections. But the microbiota adds a further complexity, and here you're looking at an egg of the parasite, and you become infected if you ingest one of these eggs, and this happens in your terminal ileum. The parasites hatch out of the eggs, and the stimulus for hatching is the microflora itself. If you antibiotic treat these animals, then you don't get strong egg hatching. So right from the start, there's an evolutionary link between the parasite and the microbiota. Whipworm, we use the mouse model of whipworm. That might be hard to see, but Trichuris murus, but it's remarkably similar to the human infecting parasite Trichuris trichura that currently infects around about 500 million people. So if you look at the gene families of these two species, one that lives in the mouse and one that lives in the human, they share over the majority of their gene families. Approximately 5,500 gene families are shared between them. And actually, if you look at the proteome of about the 6,500 proteins that are known to be shared, they show over 79% homology, so it's one-to-one ortholog. So they're very closely related, and we believe by studying the mouse parasite, we're going to learn a lot about the humans. The interesting thing in terms of commensals is that if we look down here at the bottom, this is a pie chart showing the type of bacteria families that you get in a naive animal that's not infected. We now know from a number of studies that as soon as you infect the mouse, when it becomes chronically infected, you change the microflora, and the parasite is really important for maintenance of this changed microflora. But more fascinatingly, from a biological point of view, here I'm not showing you a cross-section of the intestine. This is a cross-section of the adult worm here, a female with eggs in the uterus. Nematodes, of which this parasite is part of, have their own intestines, and if you look at the microbiota in the intestines of the parasite, not only is it different from the mouse in which it infected initially, but it's also different from the mouse in which it resides. So the parasite picks up a microbiota when it infects, changes it to its own populations, and we also know that without microbiota, the parasite can't survive. So there's a really complex interaction, and we know that the parasite presence and the response to it determines the overall phenotype that you see. So complexity is quite important, and we can dissect it using the systems that we have available to us in Manchester. We're also interested in the microbiota and the consequences of changing the microbiota. And this is work from Lizzie's lab, Lizzie who introduced us here. And this is a bit of work that will soon be published, and she's been interested in how, if you modify the intestinal microbiota, that alters your immune response. So she's carried out experiments whereby she's taken animals and treated them with antibiotics to knock down the intestinal microbiota, and then allowed them to recolonize with microbiota naturally, and looked at immune cells in the intestine before antibiotic treatment and a good period of time after recolonization with the microbiota. And she's made some fascinating observations in that the macrophage type cells that you find in the intestine are significantly changed after antibiotic treatment, even when you start to recolonize your intestine with new microbiota. And the macrophages are important in this response because you can use animals with disruptions in genes that mean you can knock out macrophages, which is shown here. But more importantly, using the variety of infection systems, you can show that these changes that are induced by antibiotic treatment subsequently alter the way that you deal with different kinds of infections. So here we're looking at bacterial infection at the top, and if you antibiotic treat and then subsequently recolonize and allow the animals to make a response to this particular bacteria, they don't deal with it as well, and this is controlled by a Th17, a particular type of T cell. And here, a completely different kind of immune response, again using whipworm. We find that animals can't control those infections as effectively as well. So these changes in the microbiota have quite serious consequences going forward and how you deal with subsequent infectious challenges. And we can look at that quite carefully here in Manchester. We're also interested in how the immune system itself controls the microflora. And Matt Hepworth, who's another Henry Dale Fellow we have here in the Institute, has a particular interest in this group of cells, innate lymphoid cells, which have become focus of many laboratories around the world over the last 10 years, and how these cells actually act to regulate the commensal microflora. And Matt is looking at how ILC3s, a particular subgroup of innate lymphoid cells, interact with T helper cells and B cells to drive IgA responses in the intestine, which are major gatekeepers of the intestinal microflora populations that we have. We can also use model systems to help understand challenges that are seen in other parts of the Institute, so in terms of barrier immunity and matrix. And here we're using other types of intestinal parasite infection to study the role of secreted matrix here, and this is mucus. So what we're looking at here is a cross-section of a lung of an infected mouse, or a mouse that has been previously infected with a parasite that lives in the intestine. And this was a really fascinating observation, where we find that if you put an infection in the intestine, you prime the whole of the mucosal surfaces around the body to make an enhanced mucus response, as shown in red and green here. More importantly, we showed that if you give an intestinal infection, but then challenge the animal much later with an infection that actually needs to traverse the lungs, as Judy mentioned, such as hookworm, the fact that you produce this mucus promotes trapping of the larvae in the lung spaces. So here we have an innate system, because we now know that this mucus response is controlled by IL-13, this key type 2 cytokine that Judy mentioned. We know that the source, or the major source of IL-13, are these innate lymphoid cells of the type 2 group, and we can show that if you get rid of the adaptive immune system in rag mice, you still get the mucus response. But if you look at these animals here that can't make an adaptive response or an ILC response, then the mucus production disappears. So we can show that now we have a new innate immune response, where you train after a primary infection at multiple sites to protect against subsequent infection by these multicellular type pathogens. And finally, just to follow on a little bit from Dave Buff, we can also use these kinds of systems to explore interactions between inflammation and immunology at one site and consequences at another. I've just mentioned that if you infect in the intestine, you can alter the mucus response at mucosal tissues. But here was some work done with Adam Dinesh and Nancy Rothwell a few years ago, where again we used this low-dose chronic infection with a parasite that lives in the large intestine to show that if you give these animals a stroke, the stroke is much worse. And you can see it by measuring damage in the brain here, in volume and blood-brain barrier damage. And pictorially, you can see the size of the stroke in these sections through the brain here, which is much worse when you have a trichuris infection. And Adam was able to go on to show this correlated with massive overexpression of this chemokine rantes in the brain tissue in an animal that's had a stroke, as shown on the right here, plus a worm infection. And in fact, if you knock down rantes expression, the damage of the stroke goes away. So by mixing together different kinds of approaches, looking at what first might seem quite an esoteric infectious agent, you can start to pick apart mechanisms that are involved in inflammation, both at sites of initiation, but also those at distant sites around the body. So I'd like to stop there. Again, thank you for listening. There we go. Thank you.